be here.
Hi, everyone. Hope everyone's having a good summer. Thank you for joining us today for the Lunch and Learn session hosted by the ASGCT. Today, we will discuss routes of administration used for gene therapies. We will include an overview of gene therapy, what are the different routes, and how do we choose the routes. This is the 6th session of the ASGCT webinar series. And as a reminder, in case you've missed any of the previous sessions or you want to go back and have a refresh, all of them are available to watch online on demand. So just go to patienteducation.asgct.org. And also on the website, you'll find an entire library of Gene Therapy 101 and disease-specific content that's free for anyone to access. My name is Vivian Fernandez. I'm an executive director of patient advocacy at REGENXBIO, and I'm a member of the ASGCT Patient Outreach Committee, and I'm your moderator for today. And today we are very, very pleased to have Dr. Kimberly Goodspeed joining us to talk about the various routes of administration that are used to deliver gene therapies. So I will give you a little background on Dr. Goodspeed. She is at UT Southwestern Medical Center, and she's been on faculty there since 2018. She's an assistant professor in the Department of Pediatrics, Neurology, and Psychiatry, and a Dedman Family Scholar in Clinical Care. She's a child neurologist and a neurodevelopmental specialist who cares for children and young adults with autism spectrum disorder and intellectual disability. She has expertise in rare genetic developmental disorders in children. That includes Angelman syndrome, Pitt-Hopkins syndrome, CLC6A1 related disorders, Phelan-McDermid syndrome, and Rett. Dr. Goodspeed earned her medical degree at the UT Health Science Center in Houston and completed residencies in pediatrics and neurodevelopmental disabilities at UT Southwestern. She is board certified in pediatrics, neurology, and neurodevelopmental disabilities. Her research, she does a lot of research, seeking to better understand rare genetic developmental disorders in children, genetic causes of developmental delays in children with autism spectrum disorder and intellectual disability, works really closely with basic scientists to help develop novel treatments such as gene therapy for these conditions that she's so passionate about. Thank you, Dr. Goodspeed, for joining us today to give the presentation. And before I turn it over to you, I just wanted to remind everyone that anytime during Dr. Goodspeed's presentation or after, you can submit questions online through the Q&A feature of Zoom. So we'll try to get to as many questions as we can during the Q&A after the presentation. So Dr. Goodspeed, I'll turn it over to you.
Thanks so much for the great introduction, Vivian. And just double checking that we can hear me and see me, but today we're going to go through a number of different routes of administration. So thinking about when we're talking about a gene therapy that needs to be given to a patient, how do we get it from that vial and into the body? So we're going to level set a little bit at the beginning just to say, you know, what is gene therapy? What do we mean when we say that? And then a broad overview of how we choose the different routes of administration, and then go into the specifics of what are each of those different routes. So what's gene therapy? We're going to start with thinking about the difference between in vivo gene therapy and ex vivo gene therapy. This has been covered a couple of times, I think especially back in like the February Lunch and Learn of this year, but just so everybody's on the same page, When we talk about in vivo gene therapy here on the left side of the figure, the vector or the viruses are conditioned in the lab to carry the healthy copy of the human gene. They are packaged in some way and delivered to the patient. And we'll talk about all those different ways that they can go directly into the patient through the bloodstream, into the cerebrospinal fluid, and so forth. But in ex vivo gene therapy, we're talking about therapy where cells are taken out of the patient as an initial step. They're taken into the lab, treated with gene therapy in the lab, and then those treated cells are put back into the patient. Importantly, though, both of these are going to require some degree of immunosuppression. So for our in vivo gene therapy, there's going to be some immunosuppressive regimen that was used in the clinical trial or is being explored in the clinical trials. And for the ex vivo gene therapy, there's going to be a conditioning regimen where you have to really prepare the body to be able to accept all those cells back into the body. So diving a little bit deeper into ex vivo gene therapy, because we're not going to touch on it a whole lot more in this talk, so I wanted to give it a little bit of time here at the beginning. Before you receive ex vivo gene therapy, your body has to undergo some degree of conditioning. What do we mean by conditioning? That's treatment with chemotherapy or radiation. And it's a way to suppress the body's immune system and make room in the bone marrow so that all of those new treated cells have somewhere to go and live. These conditioning regimens will vary. Some are more intense, some are less intense, and they're always looking for ways to make this more tolerable for patients and less toxic. But as that reminder again, we take our cells out of the patient, they're treated in the lab with the gene therapy, and then the patient undergoes the conditioning before the transplant happens. And so this is where we'll kind of leave ex vivo gene therapy. We'll have one more little part where we'll talk about it, but for the variety, most of the talk for today, we're going to be talking about in vivo gene therapy approaches. Now, just a quick overview as a reminder, what happens when you get a gene therapy? I think lots of us know that it's a one-time treatment, or we talk about it being a one-time treatment, meaning we can't redose it, but that doesn't mean that you're only going to see your doctor that gave it to you one time. So there is a bit of, logistics that go into planning and receiving a gene therapy. First, you probably are going to have to travel to a specialty center. This isn't something that you're going to be able to pick up at CVS or likely go to your family physician that's local to have it to receive it. You're probably going to have to go to a specialty center that's specially trained in administering this type of therapy. And not all gene therapies may be offered in all the centers. Then you're going to have to go through that immunosuppression or conditioning regimen right around the time that you arrive at that center or before you receive your therapy. You're going to get your infusion, however that's going to get into your body. And then you're probably going to have to stay for some degree of monitoring. We'll talk a little bit about this too, but gene therapy causes a pretty significant immune response because we're putting a lot of viral or vector particles into your body that revs up your immune system and puts it on high alert. And so there's a lot of things that we need to monitor for inflammation, make sure you're staying safe around the time of that therapy. For lots of the clinical trials, that could mean staying for several weeks or possibly several months, very close to the site of your trial. That'll depend on each different trial. But even after that initial period, you're probably going to need to go back to that site in person or at least by telehealth a few times. And there's probably going to be some long-term monitoring that, these are still early days of gene therapy, and we really want to be able to say, how do patients look in 5, 10, 15, 20 years after they've received their dose? What went wrong? What symptoms did they have? What side effects? But also, how are they doing? Have they had a lot of improvements? There's so much we don't know yet, and so that monitoring is a huge piece and really important to remember that even if it's a one-time treatment, you're still going to have a lot of connection with that site and with your physicians taking care of you. Okay, with all that preamble, let's start to dive into the different routes of administration. We tried to break this down into, first the different fluids in the body where you can administer this. So the bloodstream or the cerebrospinal fluid, or CSF, you'll see me abbreviate that a lot through this presentation. And then finally, we'll wrap up with some direct injections into different organ systems. So how do we pick the route? So this is just a broad overview to get our feet wet, but there's a lot of different ways that we can get gene therapy into the body, and it depends on a lot of different things. This is by no means a comprehensive list, but these are some of the things that we start thinking about as we're working with our basic science colleagues and starting to think about that clinical development plan. Are we thinking about an in vivo or an ex vivo gene therapy? I mentioned already ex vivo can really only be given by the bloodstream, so an IV route for now, but in vivo, we have a lot of different options. So which organs are most affected? Do you need to get it into the liver, or do you want to avoid getting it into the liver because you want more to go to the brain and the spinal cord? So these are all things that can kind of come into play as we're thinking about the pros and cons of all the different ways to administer it. And importantly, what was used in the preclinical studies? So last month, you had a really great lunch and learn about preclinical studies and how that helps us understand dosing and delivery. And so that's where we get a lot of our early cues. One, is the preclinical method going to be feasible in human patients? And some of that feasibility will be, what dose do we think we're going to use? Do we need to use a really big dose and we don't think we could even make enough to give it IV, or would it be too much to administer into the spinal fluid? Are we using adults or children or babies? And is it dosed by their weight? Because that will also factor into how much we're going to have to administer. How old are most of the patients? So thinking about body size. So a baby's very small, an adult is very large. And if we're dosing it based on weight, the baby doses are going to be smaller. And do they have any other medical problems? So one of the examples that we'll go through today highlights that sometimes if you have bony deformities or malformations or scoliosis, that may hinder some of the routes that we can pick and may help us choose or force us to choose other routes. So now we're going to start going through each of those different sections and each of the different routes. So we'll start kind of least invasive to most invasive, and then we're going to wrap up with the direct injections to organs. So first, blood. So going through the intravenous route, directly putting the gene therapy into the bloodstream. This is our IV route. And I think a lot of people are probably familiar with having a blood draw done, or maybe receiving a blood infusion or an infusion of medication through an IV. It's pretty easy to administer. Most sites are capable of putting an IV in somebody. It's pretty standard. And it also distributes very broadly. So if we know that we have a condition that we want to treat lots of different parts of the body, then this might be the route that we need to go. It can be used for in vivo or ex vivo approaches, so that's another plus. It's got some flexibility there. But some of the downsides that may or may not be downsides depending on the program, we do have to overcome this interaction with neutralizing antibodies, which we'll talk about in just a second. You're probably going to have some off-target effects again because it's going broadly. You can't really control where it's going to go. The blood goes everywhere in the body. But it may not get into the CNS or the central nervous system and brain and spinal cord quite as easily from the blood as if we're putting it into the cerebrospinal fluid. Tends to require a pretty large quantity of vector, and that has to do with those neutralizing antibodies, and can cause a pretty strong immune response because lots of our immune cells are also circulating in the bloodstream, and they're on high alert when they see that much virus coming at them. So what's a neutralizing antibody? You may have heard this term come around, and it's really just kind of the body's natural response to a virus. So in particular, in this slide, it's talking about Adeno-Associated Virus. And we've already had a lunch and learn about the different vectors. This could be true for really any vector. We're just using AAV here as an example. But a large portion of the population has been exposed to AAV in the past. So whenever your body is exposed to something foreign, like a virus or a bacteria, it remembers it, and it can make these antibodies that help it remember. So part of how vaccines work is by priming your body to recognize something foreign. But in our case, it's recognizing something as foreign that we really want to get into the cells. So our viruses have evolved and are really good at infecting cells, but our immune system is also really good at recognizing viruses and putting a stop to that. So when we think about having to overcome, you need to be able to have so much vector infused that you surpass all of the antibodies that are there in the system. And so that's part of why you have to have such a large volume or large dose in an IV route typically. And then as an example, I'm going to try to pull in an example for each one of these different modes of administration or routes of administration. I think a lot of people have heard of Zolgensma and are really excited about it. The generic name for it is there, and it was the first gene transfer therapy that was approved for a pediatric neurogenetic disorder. It's treating spinal muscular atrophy, or SMA, by giving the healthy copy, and in this initial study that got approved, they gave it by the IV route. And it was shown to stop or slow the progression. So these patients typically have degenerative muscular weakness, so progressive muscular weakness, and it was pretty well tolerated and really stopped that progression in the clinical trials. Now, as I mentioned earlier, lots of different routes might be good for lots of different reasons. And so folks are still looking at what's the best route for SMA and considering if there's another route that might be good as well. Either alone or in combination. So much is still unknown and there's a lot of work being done. Okay, so we're going to transition from the blood now to the brain and spinal cord and the fluid that sits around those organs. So that fluid is called the cerebral spinal fluid, or CSF. And there's a couple of different ways to get into the CSF that we'll go through now. So how do we get into the nervous system? I think it's good to remember how many layers we actually have to get through. So there's lots and lots of layers. They're depicted here. So we've got our skin and our bone or our skull, and then some interstitial tissue between those. But then we also have the covering or the layers of the brain, the layers that cover the brain called the meninges. And then in these blue spaces is where you will see that spinal fluid or CSF. The meninges are between the skull and the brain, and their job is really to be another layer of protection, another layer of coating, partly from kind of physical structural protection, as keeping the brain in kind of a little water bath, but also protection of layers of protection from things like infection or invasion. And the cerebral spinal fluid will really kind of fill any space that's in and around our brain. And it's flowing and it's being made every day. And it's a dynamic fluid compartment in our brain. And this is a super technical slide, but we're going to walk through it slowly. That's kind of what we talked about just now was the meninges in the skull, and that's kind of the very physical barrier. But we also have some cellular barriers that are layered in there. So this first top part of the figure is showing the kind of a very simplified breakdown of your skull and then the meninges below. And then we have a couple of different places where there's some cellular protection as well. So if we were administering the gene therapy by the bloodstream, there is a layer of cells that cover every blood vessel. So if you can believe me, there's a blood vessel under here. That protect it and force, a filtration system that's pretty tight. So not much gets in from the blood. A lot can go out into the blood, but the brain's pretty particular about who it lets through those doors. But even when we administer directly into the CSF, we're not administering it directly into the neurons. It's still going to have to flow through some layers of cells in order to get into those brain cells, which are these little spidery looking things. And so either way, there's going to have to be some layers of cells that it gets through. But it's a little bit easier when it's coming from the CSF as compared to coming from the bloodstream. Okay, so different ways to get into the CSF. So one is the lumbar intrathecal route. So lumbar meaning lower part of the back, and intrathecal means getting through those meninges. So thecal is another word for meninges. So we're literally just going through them into the spinal fluid. And so in this case, the vector is going to be delivered directly into the spinal fluid, but we're going down at the lower part of the spine. There's a benefit that there's a lower dose needed typically than IV, and that's partly because we don't have nearly as many neutralizing antibodies floating around in our spinal fluid as we do in our bloodstream. The bloodstream is much more of that immune surveillance place, whereas the brain fluid, the cerebrospinal fluid, is much more of that special space that's kind of protected. So we don't have to go through those neutralizing antibodies that are out in the bloodstream. We're bypassing them, getting straight into the CSF. It is more invasive than IV, but not terribly so. If anybody's had an epidural around the time of pregnancy and labor, it's a similar procedure. It's something that we do very commonly in hospitals to check for things like meningitis, or if somebody's not feeling well. And it's a relatively common procedure that can be done at the bedside or under sedation. It is a little more technically challenging, though, than an IV draw. And it can be structurally challenging. So if you can imagine, somebody may have some degree of scoliosis, and that's depicted here too. You know, if you have any kind of bony deformities, it might be hard to get into that space. You can still have some off-target effects, again, because the brain lets more out than it lets in. And so you're still going to be able to get some, systemic delivery, though it won't be quite as much as if you're giving it through the IV. And you're going to get much more spine than you will brain because we're administering it so low. And so the AAV or vector of whatever choice is going to be traveling up the spinal cord to get to the brain, and you may have quite a bit of administration or take up in the spine as compared to the brain. But it is a relatively low risk procedure. This is depicting kind of how the spinal cord looks. And so these little spongy things are bones. The white part is just some tendons attaching those bones to each other. And then this is some subcutaneous fat and some skin. And so this yellow structure here in the middle is your spine, and then what we call the cauda equina, the horsetail of the spine. And so whenever we administer anything in the lumbar region, we're really going below the level of the spinal cord. So the spinal cord ends up here, and we're going in down here. And remember, everything's floating in water, CSF. And so those little nerve roots will just kind of float away. And so there's pretty low risk of any kind of damage. You may have some pain or bleeding, but it's a really low risk procedure that's done very, very commonly and routinely. And as an example of an IT, lumbar IT administration, it's giant axonal neuropathy was one of the first IT gene therapies that went into clinical trial. And this is ongoing at the NIH right now in collaboration also with Hannah's Hope Fund. But giant axonal neuropathy is a rare genetic neuromuscular disorder, and it was really exciting to see it be the first out there. Since then, a lot of groups are using this route, as well as the others that we'll talk about. So it's probably one of the more common routes that you'll hear come up in conversation. Okay, so then the next place that is being looked at is Intracesterna Magna. So the cisterna magna is this space up here. So kind of at the base of the brain, top of the spinal cord, where we've got a little bit of a fluid space. And that's better depicted here on this graph. So you see this little blue fluid space of CSF, that's the cisterna magna. Again, a lower dose than if you were giving it IV because you're avoiding those neutralizing antibodies in the blood, kind of similar to our lumbar route. But in this case, it's a bit more invasive because you have a lot of important things that live right here. So your cerebellum, your midbrain, your brainstem, kind of the breathing and respiratory centers. There's a lot of important things that live up here at the base of the brain. And so it's a bit more invasive, but not unreasonably so. And it has the benefit of getting more brain and less spinal cord. So if you have a disorder that's predominantly neuronal in the brain, then this might be an advantageous route in comparison to looking at lumbar routes. Just depends on the program. Again, you could have some structural challenges. So there are some disorders that might have some narrowing or bony deformities or even scoliosis up here that could make it even more challenging or even sometimes impossible to do. Still going to have those off-target effects because we're still going into the CSF where things can get out but not get in very easily. But really the difference between lumbar and ICM is going to be lumbar is more spine and ICM is more brain. Pros and cons for each depending on the disorder. And an interesting ICM, an example of an ICM route is in Krabbe disease. So this one's in clinical trial right now with the sponsor Passage Bio. And this is a rare neurodegenerative leukodystrophy. There can be variable presentations, and the infantile onset tends to be the most severe of the subtypes. This is actually a multi-site international study, and they're looking at the safety and efficacy of delivering the GAL-C gene, which is the gene that causes Krabbe, via an ICM injection. Okay, now ICV, this is going to be the most invasive route to get into the CSF. So ICV stands for intracerebroventricular. So I've talked a bit about the different fluid system of our brain. The ventricles are the main water sacs in the brain that hold that CSF. And so this is a picture of the main system of the ventricles, looking at it as a cross section. So if you can imagine having a whole apple and you cut it in half and you look just the middle part, that's kind of this image that we're looking at here. So these sacs sit kind of in the very central part of the brain, and they connect with all of the different fluid spaces. So where we had these fluid spaces, and you can see all the blue are all those CSF fluid spaces, these ventricles are what we're talking about now. So again, a lower dose, and you're avoiding those neutralizing antibodies, but it is a bit more invasive than the IVIT or ICM. Again, technically challenging because we are having to insert a catheter through one part of the brain to inject the vector directly into that water sac, into the ventricle. You may have some structural challenges. There's some conditions that can have thicker skulls or thinner skulls, and you are going to have to drill through that skull because remember, we have our skin, our skull, and then our meninges before we even get to the brain, and you have to go through the brain to get to the water sacs, the ventricles. So there's a lot of layers you got to go through to get to the ICV route. But in certain conditions, this may be the best route to do. And looking at this a different way, you are inserting the catheter or the injection directly through all these different layers to get to that water sac, and then it's in the water sac and still has to cross over all of the different layers of cells to get into the neurons. Again, similarly can have some off-target effects, probably less so, but certainly much more brain than spine in this case too. And looking at an example of an ICV route of administration, mucopolysaccharidosis type 1 or MPS 1 is a rare neurodegenerative lysosomal storage disorder. These kiddos can actually have some bony deformities. So this is one of those examples that I was thinking of. As part of their presentation, they can develop some pretty severe scoliosis and deformations of their spine. And so these kids can actually have some narrowing of their cisterna magna. And so part of the clinical development plan for this program was that they ended up having to switch or add rather an ICM option for some kids who could tolerate it and had the space, but an ICV option for other kids who didn't have a safe option to do the ICM. So it's one of these examples too of how we have to be really adaptable as clinicians and thinking about what's going to be the safest thing for most of our patients and what's going to be the most effective, and balancing those two things. All right, and then just a quick recap, looking back over the different routes that we've talked about going into the fluid systems of the body. So first starting here with the blood, so going the IV route, we're going to have fairly low transduction of the spinal cord, pretty low transduction of the brain. You're going to need a pretty high dose to get over those neutralizing antibodies, but a really low procedural risk. Not a big deal on that one. For ICV, we're going to have low spinal cord transduction because remember, we're going up into the brain water sacs. High brain transduction, again, because you're administering it closer to the brain, needing a lower dosage than the IV, but a relatively high procedural risk of having to go through all those different layers of brain tissue. ICM, so going at the base of the brain into the fluid compartment there, you're going to have a relatively higher compared to ICV spinal cord, much higher brain than the lumbar, and then a low dosage needed and a high procedural risk because there's a lot of important structures up there. And then for the lumbar puncture IT route, then we're going to have a high spinal cord transduction, a low brain transduction, low dose again compared to our IV route, and just a moderate procedural risk associated with that. And so as clinicians, and we're thinking about the different disorders, we're thinking about all of these things and kind of weighing them across the board, as well as thinking about what part of the brain or spinal cord or body system do we really need to target most with our therapy. All right, we're going to switch directions a little bit here and go into our direct injections into organs. Since we're stuck on a central nervous system path and I'm a child neurologist, we're going to start with the brain. And so you can directly inject gene therapy into the brain parenchyma, or brain cells or brain tissues, another way to talk about that. So literally stick it into a spot in the brain and fire away with your vector. It is the most limited of the different central nervous system delivery routes. So it's really going to be localized to that one spot where you're injecting it. The benefit is if you need to only inject one spot, you're going to get a lot of vector in that one spot. So that's a bonus. You don't have your vector being wasted in different parts of the brain or body that you didn't need it to go. You're going to need a much lower dose because you're giving a very targeted therapy. You're avoiding those neutralizing antibodies because you're not going into the bloodstream, and you're not likely to have systemic effects. So if you're trying to avoid systemic effects, this might be a good route. The potential downsides, though, it's pretty invasive. You know, again, we're sticking a catheter directly into brain tissue, meaning you have to go through all the different layers of brain tissue and regions to get to the spot that you're wanting to go. It can be technically challenging. This isn't something you're going to be able to even get in my child neurology office. This is going to be a surgical procedure. You're probably going to have some local inflammation. So again, when you inject a lot of virus into one localized region, you do have some white blood cells in the brain that are protecting it, and they can get revved up and have some inflammatory responses to that injection right there. And it doesn't travel as far. So maybe that's a good thing, but maybe you do need it to go other places and just mostly be in that spot. So that can be a pro or a con. And so in this depiction, remember, we're going skull or skin, skull, meninges, brain tissue, all the way down into the spot that you want it. So here is another schematic looking at that, of the needle coming in from the outside and going into the brain tissue. And this is another just kind of graphical representation of what those brain cells look like. So again, this is that blood-brain barrier with a capillary here, or a blood vessel. And our orange cells are those supporting astrocytic cells. And then the gray cells are our neurons, our little spidery neurons. And so your catheter is literally going into that tissue, delivering the gene therapy into that region, and then it gets taken up by all those cells in that surrounding region. And a really good example of this is AADC, or aromatic L-amino acid decarboxylase deficiency disorder. So these kiddos, it's a neurotransmitter disorder, and they present with really impaired motor control, motor dysfunction, and developmental delay. And in this case, we are injecting the AADC gene directly into the putamen on both sides of the brain. So that is a region that's depicted here. So in this photo or schematic, we're looking, imagine you cut my brain in half like this behind my face, and you're looking at it from the front side. So this is going to be the midline region. And then the right and left sides of the putamen are these little blue structures just kind of sitting in the middle there. And that's a part of the brain that's really important for motor control and planning. And so the investigators found that direct injection into these regions is really where you needed that therapy. And so this was a great option for these patients, and it's working its way through the clinical trial system. Okay, intramuscular or IM injections. In this case, the vector is directly administered into the muscle. So can be used to treat neuromuscular disorders, though there's a bit of transitioning into more systemic delivery for these conditions. And there's been a few other groups that have looked at doing this in a slightly different way. It is less invasive than the CNS route, so less invasive than going into the spinal fluid or directly into the brain. You're probably going to have a fewer off-target effects because most of it will stay pretty localized to the muscle, though there is some retrograde axonal transport. So what does that mean? So when you have your muscle, and I'm going to go back to this photo real quick. So you have your muscle fibers here, each muscle fiber is going to be connected with a peripheral nerve cell. And so you're going to have a neuromuscular junction. So when you inject it into the muscle cell, the hope is that you're actually getting the peripheral neurons to take it up. And so they take it up from their dendrite or from their axons. It travels through the axon back up to the nerve terminal and will then be spread to other nerves, kind of jumping one to one. So it is less invasive. You are going to have some spread, but not as much systemically. You can get some local inflammation, and you may need multiple injections into multiple muscles all around the same time, because again, that redosing might be an issue. And again, probably limited spread. Harder to find examples of this route because again, most neuromuscular conditions are kind of transitioning more to that systemic administration. But there were a few clinical trials ongoing that were looking at giving actually a muscle stimulating factor by gene therapy. So transferring a gene that can stimulate muscle regeneration and growth rather than transferring a gene that was specific to a particular disease. So kind of a different concept that we think about now. But they were injecting this into the muscle and seeing that it was actually being taken up by the muscle and the nerves and having some retrograde transmission, maybe even all the way up to the spinal cord and brain. Some promising results, but most groups have transitioned to administration of systemic gene transfer delivery. Okay, and we're going to wrap it up with the eyeballs. So intraocular. There's a couple different compartments within the eyeball that we can administer gene therapy to. So one is vitreal. So with our eyes, this is like a cross section of the eye. So if you took somebody's eye, cut it in half long ways. This is the iris and the cornea and the lens. So the front part of the eye. There's a big fluid compartment called the corpus vitreum, and then the optic nerve that leads back into the brain. And then this back part of the eye is the retina and all the different layers that go into it. So in one route, you may need to just inject into the vitrea, so intravitreal. And then in other routes, you may need to inject into the retina, so subretinal. And this is just an explosion of the different layers of the retina blown up so you can see them a little bit better. There's a few different layers here. But we're going underneath the retina here. So where these yellow arrows are is where the vector would be administered, versus here, the yellow arrows are showing where they would be administered into that fluid compartment. So again, a little less invasive. We're directly injecting into an eyeball, which could sound scary to some, but is less invasive than directly injecting into your brain. It gets right into the target tissue where you want the gene therapy delivered, and you have fewer off-target effects because you're not going to get a whole lot of spread outside of these compartments. Local inflammation, again, is a concern. They only will do one eye at a time because they don't want to make you blind if something happens or goes wrong with those injections. So they are administered sequentially. And there is a risk of permanent damage to the eye. These have been fairly well tolerated, but anytime you're having a surgical procedure, there's always going to be a risk of damage. And so our example here is Luxturna, which is actually an approved therapy, approved back in 2017. And in this case, you get this injected directly under the retina, so in that subretinal space. And it's to treat an inherited form of retinal disease that can cause progressive blindness. And it's delivering the RPE65 gene into this region. And again, only one eye gets treated at a time. And about 6 days after the first eye is treated, then you can come back and have the second eye done. And this is another case where you really have to go to a specialty center. So on the company's website, you'll be able to see and look for a specialty center near you. But it's a specialty ocular gene therapy center who's capable of giving this highly specialized injection. All right, so that really wraps up the different routes of administration, at least as technology stands today. And there are a lot of different ways to deliver gene therapy to patients. These were all the different individual ways that are being looked at now, But there's some really exciting work looking at, can we even combine some of these ways, routes of administration to maximize how much patients get into their system. Deciding which route is appropriate for the patients and appropriate for the disease is really complex. I've simplified it quite a bit, but just know that it's really thoughtful discussions that go into each of the disease programs, and you're constantly weighing the risks and benefits and potential efficacy. We want to have the biggest impact with the least amount of vector and in the right patient population. And so it takes a lot to think about when's the right timing, where's the right location to administer it, and what dose is going to be right. And importantly, always talk with your doctor. So you want to always go back and consider, in our case, in my case, in my child's case, what would you think? What are the risks that I'm not thinking of as a parent or as a patient myself? So always encourage folks to go back and talk to their local teams. And with that, I just want to say thank you to everybody for participating today. Thank you so much for being here and for listening to this. And I hope you share it broadly if it was helpful. And a huge thank you to all the patients who participated in all the different clinical trials that led to the couple of approved medications and who are participating in the clinical trials now. We truly cannot do any of this without the participation of the community. And I'm going to wrap with just reminding you one more time, there's so much content out there. Go take a look, see what you can find, review the prior webinars, see if there's anything interesting, share it broadly across your networks. And if there's anything you think is missing, let us know, because today's session is also going to be recorded and will be posted at a later date. We want your feedback. Was this helpful? Was this not helpful? Are there other questions, other topics that we can go into? And don't forget, there's going to be another Lunch and Learn in September. So it's going to be on September 29th. And that one's going to be looking at the pathway to an approval. Thanks, guys.
Thank you, Dr. Goodspeed. It was an excellent presentation. I think we're all a little bit smarter, have learned a little anatomy, and you
A lot of
helped us
anatomy.
clarify so many of these big terms that we keep hearing a lot in this gene therapy space: cerebral spinal fluid, intrathecal, and so on. So you did just an outstanding job of breaking it up into smaller educational pieces for us so we can make more sense of it.
Thank you.
So I think we have time for Q&A. I don't see any questions as of yet in the Q&A, so I encourage people to post their questions. We have a few minutes left, and I'm sure Dr. Goodspeed would love to address any questions people have. I will start with one, and I know you talked a lot about the different routes. Is it possible for gene therapy for a particular indication to be offered in different ways, to be delivered in different ways? Does that happen, or is it always typically one route of delivery?
No, I think that's a great point. And we're certainly not wed to any one particular route of administration. So especially when we're thinking about the routes that are a little bit more systemic. So like your IV route could be combined with any of these routes. And then we have our lumbar intrathecal and cisterna magna. It may be that certain patients do better with the lumbar approach, others do better with the ICM approach. And like with that MPS I disorder, there were some patients who couldn't have the ICM, so we had to do ICV. I say we, I wasn't actually part of it, but we is the general collective of investigators. And so, yes, we all have to be pretty flexible and consider, what is going to be the optimal route. But maybe there's a second option, or maybe there are multiple options that are equal, and it just depends on the patient. And so all of this has to be thought about in that preclinical arena as we're planning the clinical trials. And most of it's coming from what was used in those typically animal studies: mice, dogs, cats, monkeys, depending on what that animal study showed. that's what really helps us guide our decision making. And if multiple routes were used, maybe we have multiple options to use in patients.
Okay. Can I throw out another term that I don't think you mentioned? And we often hear about cross correction.
Mm-hmm.
Can you talk a little bit about that and explain it in a simple way? So we can
Absolutely.
understand?
Yeah, that's a great point, because that comes into play when we're thinking about the route that we want to choose. So cross correction is the idea that if you infect and treat one cell, that it can become this little protein making factory for other nearby neighboring cells, and it can share its wealth. So it's especially helpful in enzymatic disorders, where typically enzymes, which are used in kind of chemical reactions within the cell. Think about our lysosomal storage disorders. Many of those are enzymes that are secretable. So secretable means they can be sent out from the cell and they can go and hop into another cell nearby. And so that's this idea of cross correction, is that you can treat one cell, and then whatever protein it is, if it's a secretable protein that can be released from that cell and shared, then it can go and help its neighbor and share the protein that it's made without that neighboring cell having to have been infected with a vector.
Right. Just checking to see if we have any new questions. I think our audience is quiet today. So I'll ask another. So you talked at the end about families speaking with their doctor. Any other advice you have for them? For family members or individuals that are interested in the possibility of gene therapy, and at the end of the day, they want to make an informed decision whether gene therapy is a good option for them or their child. So what other advice would you give families to become better educated?
It's such a complex topic, and the learning curve is probably pretty steep, and you're having to grapple with all of these terms that sound so foreign. And some of the stuff is kind of normal, and some of the stuff sounds like total science fiction. And so I think that especially if you're talking with family members that aren't familiar, it can sound really scary to them of what we're talking about. These are big treatments. And so I think directing them to the ASGCT patient-centered resources is a great starting point, because it really level sets everything. It helps us all use the same words, all use the same terms, understand what we're all talking about, speaking the same language. It's almost a foreign language, right? It's weird stuff that we're talking about. So I think that's a great resource, and it can be spread to your family too, so that you've got some materials to be like, this is what we're talking about. But then the other thing I think is just talking with the family organization. So many rare diseases have wonderful, robust, well-organized family foundations, and they really have their finger on the pulse of what the different researchers are doing, what's in development, who are the different players, the different researchers or company partners that are helping. And they can really help be a guide as well of what's out there, what's coming, and what may or may not be right for you and your family.
I agree. I think the patient advocacy groups are a great resource, and even other families who may have gone before them and already participated in trials.
You know, with that thought, it may even help to look, maybe you have MPS type 2, but you can look at the MPS 1 experience. There will be some differences, but kind of learning from each other. We don't have to do all this in a silo or in a vacuum. We're all doing it together and figuring it out. And there's some groups that are farther along than others. And so understanding what all those different steps are and pieces are is really important.
And the field of gene therapy is changing so rapidly. Even for those of us working in this space, it's hard to keep up with all of the advances.
Absolutely.
So it's a very promising area. We do have a question, so let me read that to you. Do you have to approve with FDA a new route of administration if new data becomes available during the clinical trial, or is it up to the doctor and the family? That's a great question, actually.
That's a really good question.
Yeah.
It's complicated. So anytime we're going into a clinical trial in humans, and at least with the FDA, I'm going to keep it to the FDA, and there are other, governing bodies in different parts of the world. But with the FDA, we typically, and again, I'll say typically because nothing is set in stone these days, but we typically have to go with whatever route was used in the preclinical models. And if we want to change something about it, we may have to do what people often call a bridging study. So it might be a smaller type of study that shows that, yeah, this new route, it's the same, exact same vector, exact same design, exact same therapy. We're just giving it in a different way. You still have to do a little bit of studying in the lab before you can take that to humans. It probably isn't as extensive as those initial studies were, but you do have to do some sort of a preclinical study, at least as things stand today. And again, this could be outdated in a year, who knows, but that's very common. And whenever we're giving an approved drug, we're giving it based on what was done in the clinical trial. So largely that helps us decide who is a candidate to receive that treatment. So when Zolgensma's label comes out, is what it's called, there are specifics of, what your genotype has to be, what your age has to be, and things that are put in there because that's who the drug was studied for. And so there are some safeguards around it. And if we're not going into that population, that would be considered an off-label use of that indication. Gets a little bit in the weeds there, but I think the take-home message is usually there's more studies that need to be done if you're changing the route of administration.
Can I ask you about redosing? Can we redose in gene therapy?
Again, as of today, no, not yet. And I hope that statement gets outdated sooner rather than later, because that would be ideal. But there's a lot of groups looking at it, but because we rev the immune system so much and our immune system works so well, it prevents us from being able to dose again. And it goes back to that neutralizing antibody issue. So lots of us have neutralizing antibodies already in our bloodstream that you're trying to overcome. Most of us don't have much in our CSF space, and certainly not much in your eye space, for example. But once we have given that gene therapy to the blood or the CSF especially, those levels of antibodies go way up. And so for the blood, it becomes, and for the CSF really, it becomes insurmountable. We wouldn't be able to get enough vector into a person to overcome those antibodies after we've revved the immune system so significantly. And there's also some risk that you could have a big immune response to it if you gave it again, because your body would be so primed to see that vector that it would just turn on full court press. And you can have lots of fever, myalgias, big inflammatory response. So as of today, no, but maybe one day.
And I guess the dosing, a lot of these gene therapies are being given for pediatric populations, so the dose, you would expect it to be smaller. So as that person grows, also, is that a limitation?
It's an interesting question. So it depends on the vector. And there was that lunch and learn vector that they'll go into the a deeper dive on all the different types of vectors that get used. But for most of the approaches that I was talking about today, probably AAVs are going to be the primary vector that's being used, at least as of today. For brain, AAV9, for muscle, AAV8, all kind of depends. There's new vectors being also developed. But all that to say, AAV9 especially is very good at going into cells that are no longer dividing, and they stay there. As long as the cell is there, they're there. Now, if they go into a cell that is dividing, so things like your blood cells, your skin cells, those cells divide rapidly still, and so you're going to dilute that out. So as a cell splits in two, one gets to keep the vector, the other doesn't have the vector, and that keeps happening for the dividing cells. But for our brain cells, what you have as an infant is roughly what you have, if not more, as you age. We tend to lose brain cells as we age. They die, they get old, just like other cells in our body. but roughly that's a non-dividing cell. So what you have when you're little should be roughly what you'll have when you're older.
That's a good point. And then if you have cross correction too, then that should help.
Even more. Yep.
Yeah.
I will say too, though, that that is still a question. It hasn't been proven yet because we're still early days. And so that's where I may put a plug in for those long-term follow-ups, is why that's so important, is that we want to see, are people still doing well, even if they got dosed as babies, or do new things come up as they get older?
We have a question as well. It says the presentation showed that gene therapy developers really need to involve patient advocacy groups from early stages of vector design and routes. Is it correct?
100%. Yes.
I agree.
I think it's super important.
Yeah.
And as an academic physician, I think it's great that, you know, when we are interfacing with our industry partners, more and more we're having patient advocacy experts that are at the table and are interfacing with the patient advocacy groups in the community. And we're all kind of there as part of the same team, because it is critically important. If the preclinical model is using some route that the patient group isn't going to be tolerant of, like maybe they don't feel comfortable with intraparenchymal brain injections, or maybe they feel better about lumbar or ICM, that should be something that should be taken into consideration as one of those pieces in that complex decision making that you're doing. It won't be the only voice, because I think all of us need to have our voices at the table, but 100% they should be involved.
I agree. The earlier we can involve the patient community, the better. One final question I have, and then I think unless there are no questions, we can wrap up. Can you talk a little bit about why we have so few centers either giving approved gene therapies or participating in clinical trials? We always talk about the specialty centers. Can you talk a little bit about that?
Yeah, I think one thing I may say is that, you know, you hear it takes a village to do something. This, I feel like it takes a full-blown like metroplex to administer gene therapy. They're really complicated. So, you know, there's complicated manufacturing. So those manufacturing centers are only located in certain regions because it's very specialized manufacturing that you're doing. You may not want that traveling coast to coast. And so you may have some centers that are using, you know, groups in different places. But even within a single center, you've got the physician and the patients, but you've also got nursing staff and administrative staff and research staff and pharmacy staff, all of these different players that have to come together and learn something brand new that most of them probably had never heard of until somebody said, hey, there's a gene therapy for this. And they said, what's gene therapy?
Yeah.
So you're starting from scratch a lot. Even in a specialty center like where I am at UT Southwestern, there's still people here that don't know what we're doing, and it's a foreign concept to them. And so it takes some education of the team to get everybody on board and comfortable and capable of doing a complex procedure with a very expensive medication that can only be given one time. And there's some high stakes to that. And so you want to have that as controlled as possible. And these are early days. I've said it a million times. My hope would be that this does become much more common and accessible. But I think for right now, when we're still all trying to learn from each other and learn what we're doing ourselves, in some cases, then it's better to have it in those single centers.
From a drug developer point of view, it's amazing how much work is involved and the number of organization and vendors and people, stakeholders that have to be involved in getting any single gene therapy trial, however small, off the ground. So yeah, we choose wisely because it is a lot of work for everybody.
It's a ton of work, and it's a lot of infrastructure building. Once you've built it, it really, you can be nimble between the different types of therapy often. But as an example, like we're a primarily neurologic disorder center versus like a specialty ocular center that's doing the eye injection. So it's really special skill sets that we're talking about.
Yeah, the special skill set. You want to give those gene therapies in clinical trials the best chance to succeed. So you want everything to be as perfect as possible and make sure everyone is ready.
Next month's Lunch and Learn. Ways to approval. So
yeah,
if you want to hear even more about that topic, I bet that I'll be covered a bit next month.
That would be a great topic, actually, to hear from a physician, a clinician's side of things, what it takes to get to be, study ready.
Yep.
That would be great. Well, I think we're almost at time. We don't have any new questions. Lots of great presentations. So kudos to you, Dr. Goodspeed, for, again, just doing a phenomenal job of breaking this very complex topic down into small bites that we could really understand. And we learned some anatomy. So
Awesome. Great. Super
I
fun.
think you did an
Yeah.
excellent job. And I just want to thank you on behalf of ASGCT and the Patient Outreach Committee that I am happy to be a part of, and just want to thank you again for doing a great job presenting this difficult topic, and also to everyone online who made the time to attend. We hope you found it useful. There will be, I think, a 5-question evaluation pushed out at the end of the recording, so please take a few minutes to answer the 5 questions. It really helps all of us that sit on the committee think about the topics that you want us to cover, so that we can offer good sessions in the future. And so on that note, I will conclude the session, and hope everybody has a great rest of the day, and we hope to see you back in September for the next Lunch and Learn.
Wonderful.
Thanks, everybody.
Bye,
Take
everybody.
care. Enjoy the rest of your summer.